In the United States, around 6 glioma cases are diagnosed per 100,000 people every year. That's about 0.006% of the population, equivalent to around 20,297 cases as of 2022. Although the number of cases are quite less, gliomas can be found to be fatal, with a survival rate of less than 50%. Hi everyone, my presentation is based off of examining genetic targets for glioma immunotherapy, and this presentation was mentored by Saswatha. I'd like to thank my mentor for helping me throughout my sessions, as I couldn't have done it without him. So for my contents, firstly, we're going to be discussing gliomas, so what gliomas are and the different types of gliomas. Secondly, we'll be discussing the goals for this presentation, followed by the prognosis, so how the gliomas come to be. Next, we'll be discussing what immunotherapies are and different examples of them, and then we'll be discussing the study focus, methods, and then the results. After that, it'll be followed by the conclusion and a little bit of a surprise. My name is Saswatha Chandrasekaran. I live in Prosper, Texas, and I'm a sophomore at Prosper High School. I'm interested in singing, drawing, and swimming, and in science, I'm interested in neuroscience and oncology. So I created a research paper and this presentation based off of these two interests. So my research paper will aim to be published within the next few months. So, first off, what is a tumor? Well, a tumor is defined as an abnormal mass of tissue that forms when cells grow and divide at a faster pace than expected. Tumors can be classified in two ways: benign, meaning harmless, or malignant, meaning harmful. As you can see in this picture right here, it's showing a cancerous tumor. Now, cancerous tumors are defined to be malignant. Cancer. Now, I'm sure we've all heard of this term before and are quite, if not really familiar about what it is, which refers to malignant tumor growth. But what about the term glioma? Well, gliomas are malignant tumor growth of the glial tissue in the brain or the spinal cord, which makes up the central nervous system. So now let's talk about the different types of gliomas. There are 3 types of gliomas, which we can see as shown below: astrocytomas, oligodendrogliomas, and ependymomas. Astrocytomas, as the name suggests, astro meaning star, originates in the astrocyte cells. Oligodendrogliomas make up the brain supportive tissue, which forms insulation sheaths around nerve cells. Lastly, ependymomas are tumors developed in the linings of the fluid-filled spaces of the brain. Overall, these types of tumors can range from slow-growing tumors to highly aggressive tumors, depending on several factors, including tumor grades and mRNA expressions. So mRNA expressions are basically how much a gene is expressed. So if something's overly expressed, we'll see more activation, or the gene will be doing more things. Whereas if it's less expressed, if there's less mRNA expression, you'll see that it's not as active as much, almost as if the gene's been turned off. The goals for this presentation. Firstly, we want to explore the expression levels of various immune genes in gliomas. Secondly, we aim to pinpoint which of these genes play crucial roles in glioma immunology. Lastly, our goal is to identify specific genes that could be potential targets for immunotherapy treatments. Now, here are some of the keywords that I want you guys to be familiar of throughout this presentation. Firstly, immune function. Now, immune function refers to our body's immune system protecting us from harmful substances that have the potential to make us sick. Immune suppression and activation. Now, this refers to either suppression, which is almost like decreasing, or activation, which is like increasing immune response. Lastly, cancer grade. So there are 4 cancer grades: grades 1, 2, 3, and 4. Cancer grade describes how abnormal cancer tissue looks under a microscope in comparison to healthy cells. Now, the higher the number, the more abnormal the cells look. A study of 100 participants with gliomas found that patients with high-grade gliomas, which in their case was considered to be grades 3 and 4, had a median survival time of 18 months. Now, if you notice, 18 months is quite low. The current standard treatment for high-grade gliomas includes surgical resection, which is also surgical treatment, external beam radiation therapy, and chemotherapy. However, even with these approaches, The median survival time for glioblastomas, which is the most dangerous type of glioma, was shown to be only less than 24 months. Additionally, there is an inherent risk based off of the location of the tumor, with resection possibly resulting in loss of function. Introduction to immunotherapies. So, immunotherapies work to either amplify or suppress an immune response. ACTs, ICIs, oncolytic virus therapies, cancer vaccines, and cytokine therapies all work to administrate an immune response on the body. ACTs involve the infusion of genetically engineered T cells to target and destroy tumor cells. ICIs have the potential to enhance T cell activity against glioma cells by preventing the tumor's ability to avoid immune detection. Oncolytic virus therapies use genetically modified viruses that kill glioma cells while stimulating an anti tumor immune response. Cancer vaccines stimulate the immune system to recognize glioma cells, which helps to initiate a targeted attack on them. Lastly, cytokine therapies administer cytokines to boost the immune system's ability to fight gliomas. In this study, we'll be discussing the changes in immune function concerning glioma grade. Additionally, we'll be focusing on possible glioma treatments with the potential to improve patient outcomes. And for my methods, I analyzed a total of 22 immune suppression and 10 immune activation genes. I used the GlioVis database under the adult TCGA underscore GBMLGG dataset. Firstly, I separated the genes into grades 2, 3, and 4. And as you can see right here, this chart is talking about the immune suppression marker list. The slide on the next page will be discussing the immune activation marker list. So then after that, we found the median expression levels for each gene for each grade. So for example, ID01, we found the median expression for that. And then after that, we organized them into 25% quartiles. So as you can see here, the 25% quartile, that will have the least amount of mRNA expression. Whereas if we look at the 100th percent quartile, that will have the most amount of mRNA expression. So as you can see here, it's as we go down and increase in quartile, there'll be more mRNA expression. So I did the same thing for grades 2, 3, and 4. As you can see here, it's the similar procedure that has been done for the immune activation list. And for our results, I obtained the results by finding the mRNA expressions that either increased or decreased with grade. For immune suppression, we found that 5 genes had significant variations in expression across glioma grades. Same for the 2 immune activation genes. These genes play crucial roles in immune response and prove to be important for future treatments. So, based on my findings, I found that the genes PDCD1 and LAG3 are promising targets for immunotherapy when worked in combination. The inhibition of these immune suppression genes could potentially enhance the immune system's ability to attack glioma cells. Now, this has been shown through various clinical studies, showing how PDCD1 and LAG3 inhibitors have potential in other cancers, which, in other cancers, I mean lung cancer and melanoma, which also proves that this can be effective in gliomas as well. In conclusion, this study highlights the importance of understanding the immune environment, which consists of the conditions and interactions within the environment surrounding the tumor. So, by finding the critical genes that could best play a positive role in immune response against gliomas, we can develop targeted therapeutics with the potential to improve patient outcomes and overcome tumor resistance. However, further investigation is still needed to fully explore the potential and effects of these new therapeutic targets. Now, I developed a short quiz for you guys. I just noticed that you can't enter your answers in the chat, but if you would like, you could just think about the answers. So, for the first one, which of the following is not a type of glioma? A. Astrocytomas, B. Oligodendrogliomas, C. Ependymomas, or D. Lymphomas? I'll give you a second to think about that. And the answer for this one is D. Good job for those who selected D. Then for the second one, which of the following genes were identified as promising targets for immunotherapies in gliomas? A. PDCD1 and LAG3, B. TP53 and BRCA1, C. HER2 and EGFR, or D. KRAS and MYC? It's a trick question. And for this one, the answer is A. Lastly, what does a high cancer grade indicate about the tumor cells? A, they're similar to normal cells. B, they're more abnormal and likely more aggressive. C, they are non-cancerous. Or D, they have lower mutation rates. And the answer for this one's B. Thank you so much for your attention throughout my presentation. Please let me know if you have any questions throughout my presentation at this time.
Thank you so much, Saswatha. I actually have a question for you. So in your research, it seems like you identified some genes that we could focus on. Did you learn anything about how long developing an immunotherapy takes after a gene is identified?
So I believe that I don't know the exact time frame, but it's essentially really hard to develop an immunotherapy because the environment surrounding the tumor is extremely because tumors have the ability to avoid immune detection. So it's difficult for the immune system to typically define which one's healthy, which kind of cell's healthy, and which one's cancerous. So to develop an immunotherapeutic, you need to have the immune system correctly identify which ones are healthy and which ones are cancerous. They need to correctly identify which ones are from which, so that they're not killing off healthy cells. So, although I don't know the exact time frame or how long it takes, I believe that it will take like an extremely long time. So that's why there's not as many um, immunotherapies as there are traditional therapeutics. But yeah,
It sounds like it's a really promising and kind of new field. So thank you so much for sharing about it.
thank you so much for having me. Thank you.